everyone. What I'm going to be talking about today is what everyone needs to know about the COVID-19 test now. And I mean everyone, literally everyone in the world, because it is being used right now to determine all kinds of policies, health, everything around the world. And it's all based upon a test that was published in January 21st of 2020. And it's actually just recently by the World Health Organization in January 20th, they said that this test, it really can't be used as a diagnostic test for COVID-19 because it has weaknesses and problems, basically is probably the reason. It doesn't adequately define it. And so why would that be? Why are we diagnosing COVID-19 everywhere? Everyone you listen to that talk about cases, the experts are saying this, we need to be doing masks and shutdowns and quarantines because of different detections and cases. It's all based upon the COVID-19 PCR test, and this is the paper that was published in January 21st in Europe by some German and other European researchers, and everything that is being done is based upon this. By the time you're done listening to this talk right now, you will know more than 99% of doctors, 99% of experts, quote unquote, and 99% of even lab officials. I've called labs and asked them, how are they running their tests? And they go, you know what? You can't ask me because it's all being automated by our machines, and those are all set in motion by the manufacturers. And we really are just, kind of running things back and forth, just making sure that the machines operate, but we're not controlling them. I even asked them, when are you changing different aspects? They go, we don't change it. And we don't even know when they were changed. And so it's all based upon this paper. And now we're told that whatever you've heard about diagnosis is not accurate. And how can that possibly be? Well, what I'm going to be doing today is explain and this paper is referred to as the Corman and Drosten paper because the first and the last author are Corman and Drosten. And this is a review of that paper done by a group of scientists from around the world that saw problems with this paper, the original paper, and recommended that the paper no longer be published, that it be withdrawn. And I'll give you the response of this journal to this review article and what they say. And it'll give you great insight into the quality of science that's being used here, and it's incredibly important. And I must say, I have read thousands and thousands of scientific papers, and this, bar none, is the worst science I've ever seen in my life. And I mean that. The worst published science I've ever seen. And this science, this paper is being used to detect, to derive all kinds of policies all over the world based on COVID-19, based on the COVID PCR test. So what's the problem? And by the way, I would suggest that you pass this information on to your doctor, your legislators, everyone else, because very few people understand what I'm about to explain here. And because basically the diagnostic test is not able to diagnose. Well, why would that be? What this review article said by these researchers, and let me tell you who these people are. They're very highly qualified by example, and I'll have all of these posted up in the comments section. There are some very respected researchers. For example, one gentleman headed the Human Genome Project at MIT, and he has patents on the PCR test with human sequencing, etc. Very experienced. Another physician used to work with Dr. Fauci, so very familiar with all of that work at the National Allergy Institute. And also another one headed up Pfizer, was a chief scientific officer for Pfizer in UK formerly, professors of immunology, etc., etc. So there's some very high quality people who were behind that. And one of the major points, I'm just going to be explaining as we go along, one of the major points is that there are 10 fatal errors with the COVID-19 test. 10 fatal errors which render the test, in their words, useless. A useless test in their mind. And they demanded, really, to have the original study withdrawn from publication. And I'll let you know their response. After that, I'll give you some indication about whether or not what my thoughts are about the vaccine, And then where our focus really should be now, instead of on this one disease where it really should be, and I've got a solution for it. I've developed a program that I'll help you with later. But let's cover the deficiencies of this test. And they said that there are 10 major flaws, if you look at the article, and I'll make it available for you with this test. And so number one, what is the number one flaw? The number one flaw is that the COVID-19 test isn't looking for SARS-CoV-2, the virus that causes COVID-19. You're kidding. It's not looking for it. How can that possibly be a test for COVID-19 when it's not looking for SARS-CoV-2? What the researchers said, they had to rush everything through in January 21st. Well, I'm not sure why they needed to rush everything through, because there were only 6 cases in the whole world. And they said, oh, it's going to be a worldwide situation. We need to rush this through. Well, how did they know they needed to rush it through when there were only 6 cases known in the whole world at that time? And so what they did was they said, we don't have any SARS-CoV-2 virus RNA available today, so we need to just actually come up with a synthetic format that we're going to be using. And they also got a format and some virus RNA from China. Now, we know now that China did have SARS-CoV-2 in January. Why wasn't that given to them? Why wasn't that the test? But right now, the way it is that it doesn't check for SARS-CoV-2, and they said because we didn't have any virus material available. And so what we did is we used silico or theoretical sequencing, which is still used today. So they're not using SARS-CoV-2, they're testing for something that is not SARS-CoV-2, okay? Number 2, the test can't tell the difference between dead cells and alive cells. So if it turns positive, you don't know if it's just fragments or pieces, or if it's really someone that's got an infection that can spread to others. So people who are positive are actually being quarantined, kept from doing things, and they might just have dead viruses on them. And I'm going to give you an analogy a little bit later to help you all understand this, because this is really important for everyone to understand, so I want to make it available and easy for everyone. Number 3 is that and one of the reasons that it can't tell the difference and discriminate between a whole virus and a part of virus is because the test only looks for a part of the segment and not the whole segment. So this whole part of the RNA is not even being checked. And so normally what is done is you do 3 segments and you look one at this end and that end, and then you pretty much can assume, well, we've got a whole virus RNA, so it's probably alive. This test doesn't even look for, only looks for about half of it. So there are fragments. Normally there are 3 locations checked for, and this one only checks for 2. So we are finding a lot of different fragments. Now, what this test also can't tell is the difference between SARS-CoV-2 and other coronaviruses. That's another problem. This is fatal. What do I mean by that? There are 7 known coronaviruses in the world. 4 of them are what we call endemic, like everybody has them, and they cause colds, okay? Everyone here listening to me has had tons of coronaviruses in your life. Every time you've gotten a cold, it's probably been a coronavirus, okay? So everybody in the world has some coronavirus particles and RNA in their body. It's endemic. Everyone has it. We don't get rid of it. It's always there. This test can't tell the difference between SARS-CoV-2 and other coronaviruses. There are places on the genome that they could have chosen that is distinct for SARS-CoV-2 or SARS-CoV-1 that they didn't choose. Now, I said there were 7. 4 of them are endemic that cause colds. Another one is MERS, which is Middle Eastern Respiratory Syndrome, that occurred back around 2012 and has died out. And the other 2 are SARS viruses. The first one is SARS-CoV-1, which started out in 2003 and hasn't been around since 2006. And then the current pandemic. Those are basically only 7 coronaviruses that exist. This test can't distinguish SARS-CoV-2 from a cold virus. So if you've been told that you are positive, it could have been just from a cold virus, okay? Next is that there's no standard operating procedure that they gave to labs. And so So when they come out, they just say, well, here's roughly what I would recommend that you do, or what we'd recommend that you do, but there were no distinctions. In one place, they allow, for example, you can choose this base pair or this nucleotide or that one, whichever you want to do, in 6 different locations, which allows for 64 different variables in different labs in different parts of the world. And they don't give strong recommendations about how often to do other things like temperature, cycling, and how much to add things. And so what happens is labs all over the world in different countries are doing different things. That's why you're getting different results in everywhere in the world. Now, are you starting to understand why I'm starting where I said earlier, this is the worst science I've ever seen? And this is the test that everyone is using everywhere in the world to base everything that is going on with economies, shutting down businesses, everything that's occurring is based on this test. That's what's happening. Now, in another location, they have, they recommend 4 to 8 times the ingredients that is normally put in there. And the result of putting in too much, it'd be like you having a recipe and then taking just one of the ingredients and adding 8 times of what would normally be occurring in that recipe. Now, is that something that you would want? Would you expect that what's going to come out of the oven is what you're anticipating if you have one ingredient that is 8 times higher than normal? No, and that's what they're saying here, is that they're recommending 4 to 8 times the normal ingredients. They're also recommending the wrong temperature and excessive numbers of cycles. The wrong temperature renders the outside, from the review, they say that it renders everything useless. If you don't have everything at the right temperature, these are very sensitive tests. If you don't have them at the right temperature, you don't get accurate results. And then the Corman and Drosten, in the PCR test, what they're doing is they're cycling, which means multiplying virus, RNA and DNA so that the machines can read them. If you cycle them too many times, multiply them too many times, everyone becomes positive. In the test, they recommend 45 cycles, okay? Normal is not over 30. And in fact, at 35 to 36, what you get are just dead parts and pieces. And this fall, all over the United States, they were cycling at 38. I asked labs, when did you switch it up to 38? And they go, we didn't switch it. The manufacturers of the machine switched it. We had no control over it. Even Dr. Fauci said last summer in a podcast that he was on that anything above 35, 36, and I've got on here where in that you need to listen to it. He even says anything above 35 or 36 is just dead nucleotides. It's just bits and pieces. It's meaningless. And yet hospitals all over America were doing it at 38. And so there's no validation, no controls. And by the way, there was a conflict of interest. When the original paper was published, they said there's no conflict of interest, but actually when it was published, two of the authors were on the board of the journal that passed it, and actually they submitted it on the 20th, January 20th, and it was accepted on the January 21st. And they even admit that they only looked at it for a few hours. In other words, what it looks like is that just because those guys were on the journal is why it was passed. It really wasn't reviewed. And then they said there was no conflict of interest. Well, there was. I think at least 4 of the authors are connected to a PCR test that was actually already produced based on this study and actually already in labs around Europe. It had already been dispensed before the letter was even published. And there's also a connection to another lab. So obviously it looks like there was some money involved. And so from the review, they said, this is a useless test. And that's what we're using all around the world. Everyone is scared based upon a test that is useless. So how did the authors of that journal respond? How did they respond? They responded by saying, well, basically we brought in 2 other experts. They didn't say who they were. They said it was fine, so we're going to keep it published. We're not going to do anything. They didn't address any of these 10 points. So in the scientific world, that's like saying, get lost, forget it, drop dead. I don't care what you want. We're not changing. That's what's going on here. A true scientist would say, you know what, you brought us some good points. Let us address them. Let us change these parameters so we can make the test better. They didn't. And that gives me an indication of the quality of the science. It's horrible, they know it, they had no response to it, and they're keeping going. So every time today when you hear in the news somebody saying, oh, the test positives went from 9.7 down to 7.3, that's crazy. There's no accuracy with any of that. Or, oh, the cases are going up now, and oh, these people have, this many people, 400,000 people have died in the United States. Based on what? Based on this test, which is useless, meaningless, has no scientific validation. And so, let me give you a quick analogy. What we all need to do is stop and think for ourselves and stop basing everything you're doing on fear. Let's say, for example, let me give you an analogy to help bring all of this stuff together, and I'll bring some of these points. It's not a perfect analogy, but it'll help you understand, okay? And I don't know if you know this, but cougars, when they get old, what they'll do is they'll, when they get too old, they can't chase down deer and small animals, and so they'll go near humans and human population and hunt humans, because they can run down humans if they can't run down a deer. And unfortunately, that occurs in certain areas, and maybe you've heard of that. Well, let's say, for example, there was an old cougar, and he came down to close to a city, into a park, and there was 100,000 people near that park. And he found 2 or 3 people, and he killed them. And it was a popular park that everybody had been to, and so everyone freaked. Everyone thought, oh my gosh, cougars are now killing people in the parks, and we don't even know how many cougars there are. We don't know how bad this is. Oh my gosh, everyone in the city might die. We better get to the bottom of this. Well, unbeknownst to them, what happened was that cougar did eventually die. Scavenger animals came, they tore it to pieces and took its DNA and put it all over the park, and it was a very popular park, and thousands of people from the city went through the park. So the people demanded, out of fear, that some organization figure out how bad the problem was. So they came up with a genetic test, and that genetic test, and that organization who came up with it was the Center for Cougar Control, the CCC. And you know, they didn't have any cougar DNA, so they asked from another country, they asked for some cougar DNA, and so they went to maybe like a foreign country and said, can you give us some cougar DNA? And they thought, well, you know, we don't really have cougars here. And they contacted the Center for Cougar Patrol in another country, and they said, you know, actually, we don't have any cougars here, but we have different cats like tigers and stuff, so we'll send you some DNA over that you guys can use to make your test, because it's probably close enough. And so what they did is they came back and they started producing that DNA test, and they started testing everyone's shoes, because they wanted to find out how bad the cougar problem was. And they started testing shoes, and they found out that, oh my gosh, everybody in town, there were thousands of people who had cougar DNA on their feet. And so it came out in the news that there were thousands of cougars that are in the forest, and everyone became even more scared. And so then they started to realize that, wow, the test doesn't differentiate between house cats and cougars or tigers. And so they started testing people in homes who had house cats, and they found out that everyone that owned a cat had a positive test for a DNA. So those people were called super spreaders. And so they got super scared with those people. And so then they quarantined all of those super spreaders, and they put them in. And they recommend everybody in town put boots on, because, oh my gosh, because we need to be protected from this cougar DNA, because for some reason people are dying of this cougar DNA. They went to the hospital, checked the shoes of people who had just died in the hospital, and by golly, they said, you've died of cougar DNA because we found it. And so people kept dying in the hospitals with cougar DNA on their shoes. And so everybody panicked, and so they started going on and on and on, but they didn't have cougar really DNA. They couldn't tell the difference between dead and alive DNA. They couldn't tell the difference from other types of cats. They didn't have any standard operating procedures, so they recommended and put everybody, and that it just got absolutely out of control, and that's very close. It's not an exact analogy, but it's very similar to what's going on today. So, in light of all of that, in light of a worthless test, are you going to get a vaccine? Well, let me tell you about the vaccine a little bit. The vaccine, for example, there's 2 that are out today, Pfizer and Moderna. Moderna has never made a vaccine before. That's kind of interesting. And it's an experimental type, and they're calling it a vaccine, but it used to be a therapy for cancer treatments. And instead of injecting dead viruses, etc., so that your own immune system can form a response. They're injecting messenger RNA so that your cells keep making an antigen or an allergy type thing, so your immune system will respond to it, and so it leads to kind of increased inflammation continuously, and it just is going to keep making it. So it's an experimental type of vaccine. I'm not sure it really fits into the category of vaccine, but people are saying that it does. It's never passed animal tests before. This kind of technology has been around for decades, but it's never passed animal tests before. All of the animals that had ever been tried for died, and they didn't die immediately. They died later when they were exposed to a very similar virus. So why didn't we do the animal testing this time? We didn't do any animal testing with the Pfizer vaccine or the Moderna vaccine. We didn't, because we were in so much of a rush. We're in so much of a rush. We're just racing this out. And so no animal testing has been done on it. And so what I'd strongly recommend that you do, instead of listening to a doctor on TV or the press, instead of getting in line and racing down there, that you talk to your doctor, specifically someone who knows you, as to whether or not you should get that vaccine. And I strongly recommend that you have your doctor listen to this tape, because there's a 99% chance that you know more than your doctor does, because they don't understand this Corman and Drosten study. They don't know how it was structured. They're trusting the lab companies, they're trusting the hospitals, they're trusting the government, they're trusting everybody, and nobody has checked into this, and they should. So what do we do now? Where should our focus be? Should it be on a disease like this that has a test that's completely useless and unreliable, according to the review authors? Or should we be focused on what we really need to do to take care of our health? Because to protect your health in the future, you need to have a healthy body. And I'm not talking about a few pounds of weight loss. I'm talking about totally transforming every cell of your body. Obesity is still climbing. Diabetes is still climbing. And all of these issues are still very significant, and we need to be focused on that, not running away from something that we have absolutely no knowledge how far it's spread and how lethal it is, because all of those statistics are based on a useless test, okay? So let's get back to basics. Let's get back, stop the fear, and let's get back to taking care of ourselves, enhancing our body. I know that obesity kept increasing because it happened to me personally, and I didn't know why I was gaining weight. I tried the popular diets. I thought, well, it must be working for other people. And I tried them personally, and I found that the food wasn't working. The food tasted horrible. And I thought, I'm going to go back to the drawing board. I'm going to start from scratch, create my own program. I did. And in doing so, I lost 50 pounds, and I keep it off effortlessly, because now I know what's behind all of this stuff that's causing the obesity epidemic too. And it's so complex that I couldn't do it in just like one talk or one quick phrase. This is what I'm doing. I put together 60 videos, and I call it the I-60 challenge. I put together 60 videos to walk you through a nutritional transformation, an optimal health lifestyle change transformation. That's the only way to do it. Quick diets aren't going to work. You need a transformation. And so that's what I created. So in that are 60 videos. I teach you about a minute every day for 60 days. Included in that are recipes, foods to avoid, foods to eat, and I also have included what I think is the world's finest supplement to help in that whole process, called Ignite. And I'm so impressed by that. We have worldwide exclusive to it. You can't get it anywhere else in the world. It has been shown to have 7 health benefits in clinical trials, and among them is that it doubles weight loss over diet and exercise alone, proven at a university in the United States. It also doubles adiponectin, which is a great hormone for fat metabolism and anti-inflammatory effects. And thirdly, what it also does is that it doubles AMP kinase, which is an enzyme associated with the greatest amount of longevity. There's a great research right now, and 4 other benefits besides that. So the product itself is not an anti-inflammatory, but it doubles AMP kinase, so it provides a balanced and a good, healthy inflammatory response. And so you might be thinking, well, how much would a program like this cost? $100 a month, $200 a month, $400 a month, with all of those videos and teachings? No. I want to make it as available as I could and reduce the price as much as possible. I make it available at $59.95 a month. because what I want to do is make it available for anyone. It's not about the sale, it's about transforming lives, and I guarantee it 100% with a 60-day money-back guarantee. And I have helped people in 30 different countries and spoken to audiences of thousands, given interviews all over the world in 30 different countries around the world to promote optimal health. And now it's my turn to help you. What I can guarantee is that for a year from now, your health will be different. Will your body be an asset? Will it be a benefit helping you to have the optimal health? Or will it be your greatest enemy? You make the difference, you make the change, you can make the change, and I want to help you in that process. I strongly recommend you go with the I-60 challenge, quit focusing on everything else in the world that's trying to scare you, get back to basics and improve your health. Thanks for listening.